Hello everyone, today we are going to learn about cloning vectors, some important aspects related to plasmids, cosmids, phagemids, lambda bacteriophage, and M13 bac yak mac, which are some short forms like yeast cloning vector, bacterial cloning vector, like that. So we will be learning about all these. So let's have an introduction about this topic. DNA encodes genetic information in all living cells. Genetic engineering is a branch of science that deals with the transfer of DNA between the hosts by in vitro enzymatic manipulations. So we will be conducting some enzymatic manipulations which are actually in vitro. In vitro in the sense inside the body of that. Not inside the body of that organism, outside. Okay. Inside, which will be shortly represented as in vivo. But this case is in vitro. Okay, outside. So enzymatic manipulations will be conducted. Then since most of the DNA fragments are incapable of self-replication in host cells, an additional segment of DNA capable of autonomous replication must be linked to the fragment to be cloned. This autonomously replicating fragment is known as a molecular cloning vector. That one is called molecular cloning vector, and we have different variations in that. So they facilitate the stable integration and replication of exogenous DNA inside the host system. This technology is termed as molecular cloning. It involves a series of sequential steps which include restriction digestion of DNA fragments, both target and vector DNA, then ligation of the target DNA with the vector and their subsequent introduction into the host organism for multiplication. So these are the basic steps in this process. First, we need a series of sequential steps like introduction of this restriction digestion of DNA fragments. We need both target and along with that vector DNA. So we need both the species. Pieces in the sense fragments, DNA fragments. Then ligation of the target DNA with the vector and their subsequent introduction into the host organism for multiplication. We need many copies. Okay. So in general, vectors should possess some key features necessary for their function, like capable of autonomous replication inside the host, which is represented by this short form ORI site, which is nothing else but the origin of replication. You might have already learned about that. A compatible restriction site for insertion of DNA molecules, like insert. So presence of selectable marker for screening positively transformed cells. We need selectable marker. And smaller in size and able to incorporate inserts of larger size. And types of cloning vectors we mentioned earlier, there are many types. So first one is plasmids. Plasmids are naturally occurring extrachromosomal double-stranded circular DNA molecules that can replicate autonomously inside host bacterial cell. So these are nothing else but extrachromosomal double-stranded circular DNA molecules, plasmids, which are primarily situated in case of, bacterial cells. Okay, so plasmids range from about 1 to 250 kilo base pairs in size. They are stably inherited extrachromosomally. They encode for only a very few proteins. Yeah, one word, that is the extrachromosomal inheritance, is something which you might have heard in case of chlorophyll mRNA. And DNA, extrachromosomal inheritance, is followed by that. Extrachromosomal in the sense outside. Chromatin is something which is primarily related with the DNA. And when we talk about the case of this plasmid, it follows the case of inherited extrachromosomal inheritance. So that is something which is related to extrachromosomal inheritance outside nucleus. It has some sort of independent inheritance. Like the case with chloroplast DNA. They encode for only a very few. Same is the case with mitochondrial DNA. Empty DNA is also like that. Extra chromosomal inheritance. They encode for only a very few proteins which are required for their own replication. These protein encoding genes are located very close to the origin of replication, ORI we mentioned earlier, ORI site. The rest of the needed proteins are provided by the host cell machinery itself. So some important proteins are created from the transcription and later the translation of the encoding genes that are located very close to the origin of replication. But other proteins are provided by the host cell itself, host cell machinery. There's only a small portion surrounding the ORI site is required for its replication, hence the parts of a plasmid can be detected and foreign sequences can be inserted without compromising replication process. Okay, so the number of molecules of a plasmid present in a single bacterial cell is termed as copy number. It ranges from 1 to 50, less than 50 per cell, copy number. The number of molecules of a plasmid present in a single bacterial cell, which will be definitely undergoing some fluctuations. So that thing is called copy number. So it ranges from 1 to 50 per cell. But this number is specific for given plasmid residing inside the host cell. There are different types of plasmids. You could see that with this figure. Relaxed plasmids, stringent plasmids, fertility plasmids, resistant plasmids, then degradative plasmids, virulence plasmids, which show some sort of, transforming some sort of virulence, so capable of transforming a bacterium into pathogen. Then based on ability for sexual conjugation, we learned earlier what is conjugation. So there are some plasmids which are related to this conjugation function of transfer of a factor. Conjugative plasmids contain specific transfer genes that can facilitate the process of conjugation in a bacterium. Then non-conjugative plasmids are also there. And later we'll be learning some important plasmids which I will be explaining in the next video. Thank you for listening.